Surut, for chairing this session. I'd like to invite Dr. Uday Saxena, who will be chairing the next session, to please come on stage.
Everybody. So we have 3 presenters for this session. All of them are from the industry, so fasten your seat belts. I think you're in for an exciting session. The ground rules are 30 minutes for presentation, is it? And 35. Oh, so you increased from yesterday. Okay, 35, and then how many for Q&A? 10, okay. All right. So the first speaker is somebody who I literally grew up with. I don't know where he is, but Manny Wangla is the... Our next speaker is Adrian Roth. He's going to talk about MPS to support safety assessment of immuno-modulatory drugs. He's the principal scientific director of personalized healthcare within Roche's clinical development organization. He's been global head of Roche's investigative safety department and development organization, as well as supports ADME and toxicology. He also holds a professorship at the University of Basel, Switzerland, and has authored many publications. One of his key areas of focus has been establishment of modern human cell models, such as organs on chips, organoids, MPS, which his team has introduced at Roche. His talk today, as I said before, is going to be on MPS to support safety assessment and immunomodulatory drugs. This is going to be a virtual presentation. Welcome, Dr. Roth, and please take it away. It's all yours.
Thank you very much for the kind introduction. Good morning, everyone. It's a great pleasure to be able to speak here, and apologize that I couldn't make it on site here. It would have been certainly much better to be there. I hope to be able to join next time. So as you heard in the introduction, I would like to take you to a journey from my past role in preclinical safety, supporting safety assessment of immunomodulatory drug over to clinical development and personalized medicine, which is a focus in my current role. So when we look at the use of microphysiological systems in preclinical discovery, I'm sure you've heard a lot about these aspects of drug development and the use of MPS systems. There's different areas where those systems have found great application and really have been starting to make a big impact, be it in very early phases around disease modeling, for target identification, the selection of best candidates for further investments early on, replacing simple cell line models, and then also the important step of enabling entry into human, where there's typically lots of mechanistic issues to be resolved, where it's all about the human relevance of findings in animal tests that are still typically required by regulatory authorities. Now, in a nutshell, the focus of work in this preclinical discovery stages is that you're facing a large diversity of different drug candidates, and you would like to use MPS systems that should be, in the best case, representing kind of the average human, and you want to have, with a decent throughput, you would like to be able to test a large diversity of drug candidates and see which are the best ones to further invest into. Now, when we look into the clinical side, which is a bit of a newer area for application of these types of systems, it's almost a bit a reciprocal situation. You have a huge diversity in the patient population, And now, in a nutshell, what we have been doing so far is that there's one treatment for everyone, and this may work in some, but not to the best possible extent in others. And so, with a more tailored approach, and with being able to group patients, to stratify patients, our hope is to be able to bring the best potential drug to the right patient. So in a nutshell, you can summarize what we're doing here is not testing a diversity of drug candidates, but rather a diversity of different patients. And I'm going to talk about this later on in my talk. So overall, what spans over all these different areas is our common vision. When developing these types of systems, we would like to reverse translate from a human situation to an in vitro system, to ultimately then be able to predict back from these in vitro systems to the human. So the starting point is always, or should always be, the patient. And when I say the patient, I'm relating this to the data that's available, real world data, plus ideally human tissue, patient-derived tissue. If that's available, we can go via a reverse translation approach back, build up relevant human cell models. Together with the data, we can benchmark them. If we feel those models have predictive power, we can use them again for future drug development programs to predict the human relevant situation. Now, in order to achieve this bold vision, there's a couple of different aspects where certainly everyone here and additional people in the community are working on hard. So first of all, of course, the most obvious one, there's certainly more basic R&D needed in bioengineering and cell biology to better understand what are really the important aspects to make such a complex model more physiologically relevant. And we see great progress every day, including also at events like this one here. Then, of course, data science comes into play both for the prediction and also for the reverse translation approach. This is always a tricky thing and requires lots of skills, including predictive algorithms. We can talk about AI and so on and so forth. A bit of an overlooked topic, what I keep on saying in all conferences since I'm giving talks here, is that we do also need access to patient data. Certainly, this is a sensitive topic, so this data has to be anonymized. But I think in order to really make solid predictions and solid establishment of these models, patient data is absolutely required, and of course also then the patient tissue, or depending on the type of approach chosen, stem cells from the patient. Now, in order to achieve that, we do believe that this requires cooperation and collaboration between a number of different stakeholders, spanning from academia, clinical organizations, which are coming more and more into the world of MPS. Of course, patient organizations, if you talk about rare diseases, for example, could play an important role. Data science experts, small companies, large as pharma, and also regulators. So this is a bit our vision. Clearly should tell us that conventional preclinical in vivo testing may not be relevant, or in this case here, I would say, is just not possible. So we had no option to move forward. So we realized there's an urgent need for novel tools to assess the pharmacology and toxicology of these new types of drug candidates. And a couple of years ago, that was for upper management, was really the call for action to start and invest big time into these types of models. Now, I would like to go into an example which was among the key drivers for establishing these types of models, and is also related to the title of the talk, Cancer Immunotherapy, Immune-Modulatory Drug. So one of the challenges there, despite the big aspirations and also great successes that we've seen, is that still nowadays only a limited number of patients actually do respond, and we don't have that much of a good handle and understanding why and which are those patients that actually respond and which are those that do not respond. At the same time, and that was relevant for us both in preclinical safety and now also in clinical safety, we do have a relatively high rate of adverse events, and we don't have good biomarkers to predict them. And you can see on the left-hand side, these adverse events actually happen all over the place. It's not confined to one particular area, one particular organ. They seem to be happening just basically everywhere. Now, as a side comment, and I'll come to that later on, is what has been a notion I would say 2-3 years ago, is that about 70% of patients that were discontinued due to these adverse events actually show a strong treatment response. So there seems to be even a link in this case between the drug response and the risk for adverse events, and I'll come to that later on. But let's first just stay with the preclinical safety assessment. When we look again at clinical toxicities with the first CIT drugs, cancer immunotherapy drugs, here 2 famous examples. These are so-called checkpoint inhibitors, ipilimumab and nivolumab. You can see here from a little bit of an older publication, the different types of toxicities, the grades, and the onset. So again, underlining, by no means this is confined to a few target organs. It's all over the place, and that's something that was very initially, that was certainly unexpected. Classic conventional antibody types of drugs were believed to be almost devoid of any kind of side effects. But when we started working with these types of highly engineered immune-engaging molecules, the side effects came, and they came quite significantly. So we set out to establish cell models for preclinical safety, a whole suite of cell models to be able to address what we call on-target off-tumor safety liabilities of these bispecific antibodies. The way they're set up is typically, not in all cases, but typically primary human tissue polarized where required. We would add PBMCs, or at least aspects of a component of the circulating blood cells, all tissues and cell components from the same donor, or at least, if not possible, from the same donor, HLA matched. And so this is then how it would look like. You have the intended target tissue, you have an extracellular matrix, and you have the blood compartment typically consisting of an endothelial tube plus PBMCs. And that, I think, is clear to everyone here in the audience. So current conventional models would certainly lack these essential biochemical and biophysical factors found in the native organ. So this is clearly a rationale why investing in these types of models. And our goal was really to be able to recreate drug-induced, first of all, drug-induced activation of immune cells, the initiation of downstream events, including the cell migration to the site of action. And most importantly, at the site of action, we would like to have a measurable effect that allows drawing conclusions on the drug molecule properties, the desired and the undesired effects, and most importantly, the relevance to the patient population. So quite a few steps and quite a challenge what we wanted to achieve with these types of models, but that was really our motivation. So when you start these types of programs, you need to think of a number of different technical aspects. I alluded to these 3 different steps, and you see here a couple of examples. This is already probably already a bit outdated. These were first attempts, examples from the labs where people have been working on these different steps, from immune cell activation to targeting healthy tissue, and in the end, being able to measure an adverse event in the healthy tissue. This may sound quite obvious, but in some cases, this is not so trivial, and you need to tweak and optimize the system quite a bit, especially in the early days when these data have been generated and we were simply lacking any kind of benchmark data and didn't know how to do that and what was the necessary, I don't know, diameter of the channel and how many cells do we need there and so on and so forth. Still now, when preclinical teams are developing these models, you can see occasionally an effect. Let's say you measure an activation of some immune signal, you can measure maybe some cytokine release, but you don't see in the end what might be the consequence, yeah? Which is a potential adverse effect. So you're not really sure, is this now just some activation or is it actually something which really could be clinically relevant and where teams should pay attention and maybe address that during the course of the drug development program. So a lot of optimization work the team at that time has been doing. The questions we wanted to focus on with these type of models were clearly the so-called on-target off-tumor toxicities. There's other types of adverse events that are important to be addressed in preclinical safety, for example, for these T-cell engaging molecules, but also if you think about CAR-T and related areas. So in today's talk, I'm not going to focus on all these cytokine-mediated toxicities. Also off-target is maybe something that is a bit less of a priority for these types of molecules and is also being looked at via completely different approaches. Again, also then autoimmunity infections, inflammation, and these things which are related to probably more the adaptive immune system is something we're looking at via different approaches. But our intent here, our use case for MPS was really on target of tumor toxicities. So the tumor target, which is in any case expressed also in other parts of the human body, in the healthy tissue. Is this a concern for an early program or not? And how can we address it? What's our window of opportunity? Can we measure it? Can we assess it? Can we use such systems to improve the first candidate molecules? So what I would like to show you is a few examples from this relatively early work that we have been doing. In this case, this is stems from a multi-year collaboration with Boston company Emulate, using their, at that time, new system for the lung and later on for the intestine. I'm showing you here real portfolio examples. So this was a T-cell bispecific that would target the CD3 as the immune cell target and folate receptor 1. The molecule has been shown to be efficacious in human breast cancer, patient-derived xenograft models. And unfortunately, one of the earliest in vivo studies in cynomolgus monkey, there was severe on-target toxicity in the lung. Now, as you can see on the left-hand side, based on available tissue slices, it was known that actually the target is expressed on the inner side of the lung, so on the apical side of the lung epithelium. But as there is this epithelial barrier, it was believed that shouldn't be a concern because the molecule would not reach the target. And so we had to learn the hard way that actually this is not the case. And so the team felt before we go into a next round of in vivo study, could we make use of Emulate's tissue chip model in order to test different variants of the molecule. So this is, for those who are not familiar with this type of organ chip model, this is how it looks like. So it has two channels. The top channel consists of epithelial cells. There's a porous membrane, and below you have endothelial cells, where we would be sending blood cells backwards and forth. As a first test, we would be looking for the expression of the target, of course, and as you can see, it's nicely expressed on the epithelial layer in this chip, which was very reassuring. Now, as you can imagine, a lot of different types of assays, trial and error needed to be carried out. I'm really showing you here a very tiny amount of some of the latest data coming out of this chip. First of all, what we could then finally measure is when adding the molecule, and as you can see, we had two types of molecules. We had a non-targeting one as a negative control, plus a strongly target-engaging T-cell bispecific. We tested with and without the addition of PBMCs, and it was very reassuring and very encouraging to see that we could also actually measure caspase positive cells collected on live chips when both PBMCs were present plus the targeting antibody. This was further confirmed then actually when looking in the epithelial channel where the target is expressed, folate 1 receptor, we would see an accumulation of primarily CD3 positive T cells, further confirming that actually what we're measuring here is really a specific effect that very much follows the intended use and mode of action of a T cell bispecific and not just some basic random measurement, what we're doing here. And this is underlined here, as you can see here again, when adding both PBMCs and the targeting antibody, you see a strong increase in CD3 positive PBMCs. So having all of this measured, we could start going back to the team and say, okay, now we can use the model to test different types of molecules. Here showing with a quite extreme example, we had a very low targeting folate 1 receptor molecule and a strongly target one that was used for the validation of the model. So the idea here was really to use the model to identify a safer molecule. And as you can see with this type of experiment here, actually the window of opportunity until safety effects start to kick in is much better with an alternative molecule than with the original one. So that was our use case for this lung model here. Now switching over to a very related example here, again, T-cell bispecific, this time a T-cell bispecific that is targeting carcinoembryonic antigen. This is a very well-known tumor target. It's known to be expressed in the gastrointestinal tract, and so we were interested in looking into this again using the emulate chip model. And as you can see on the left-hand side, it's well known based on human histopathology that the expression of the target is much stronger in the colon as compared to the duodenum. And when we were looking into the intestine chip model, it was actually nice to see that we could recapitulate this difference in target expression on an in vitro level. So that was already a very good start. Now, again, optimizing the model and doing lots of rounds of trial and error, we could then finally start generating data which looked quite encouraging. So first of all, we wanted to see a molecule-specific response. And as you can see here in this example, by measuring the release of cytokines, as expected, the high affinity T-cell bispecific exerts more pronounced cytokine release than the low affinity T cell bispecific. So this is obviously what we wanted to see. Now we started to combine it with the region-specific response, and as you can see very nicely, and again, as expected, we do see stronger effects in the colon versus the duodenum. In this case, we could also see the presence of T cells plus cytokine release. What's shown here in this graph are the T cells, but we also measured it and confirmed it by cytokine release. So very encouraging first data that told us, yes, actually these complex signaling pathways that a T cell bispecific drug has, there's actually an opportunity to recapitulate that using these types of complex models. Now, switching gears a bit into the disease model side. So very quickly, this is a relatively also among the first work we did together with Mimetas. So this was about recapitulating aspects of inflammatory bowel disease using the Mimetas chip model. So a gut on a chip model of intestinal inflammation. So the major,
last 5 minutes
idea
for you.
here.
Excuse me, Adrian. Sorry to bother you. Last 5 minutes.
Okay.
Thank you.
All right. So I will then really move forward quickly. So in essence here, the major idea was really to be able to see migration. So first of all, the recruitment of activated immune cells and the migration to the sites of target. And this is what we could measure here in that model, and we were very happy about that. So in the interest of time, I will skip the other example and just for the last couple of minutes tell you a little bit about our ambition, what we want to do in clinical development, where we really see microphysiological systems as a potential window into our patients' individual physiology. And how can we use that? So we would like to use them, patient-derived models to predict IRAEs in trials, and also identify patients at risk, develop mitigation strategies early on. So this comes together here with some of the established tools in clinical safety, looking into genetics, making use of real-world data. Digital tools have become really important to monitor patients as kind of a new kid on the block, patient-derived tissue models. Now, again, one of the starting points has been CIT, where we see there's a certain link between the survival and the occurrence of adverse events. We also saw that in our own trial. This is Tecentriq, a checkpoint inhibitor, where we saw that the appearance of low-grade skin adverse events, drug-induced adverse events, comes with higher chances of survival. And furthermore, what we could also demonstrate is that there seems to be a genetic predisposition for the occurrence of skin adverse events, which is underlying this. And so we asked ourselves, can we use patient cells before they're actually treated to predict if there's a risk for immune-related adverse events? And can we use then such an approach to implement mitigation strategies early on and address toxicity while maintaining higher efficacy? In order to achieve that, we were looking for partners that could help us with a new type of tissue model where next to all the scientific criteria that we're typically discussing, you have to think of other aspects, operational, technical aspects that meet the aspects of a clinical trial. So minimal starting material, an easy automatable workflow. It has to be small, scalable, and fit into a decentralized clinical trial setting. And of course, a very short biopsy to assay time, and you need informed consent, and so on and so forth. So we ended up working with an approach that requires only about 1 week from resection of a very small skin biopsy and ending up with plates of skin organoids to be able to be moved forward to testing. So this is what we're currently doing, and our goal is really to test the patient's organoid and then define individual treatments and define individual measures in terms of safety. And this very much should fit into current algorithms around personalized medicine programs. You probably have heard, for example, of the molecular tumor board, which is here listed as a key element, one of the newer key elements of personalized medicine programs. And you can see here from a recent publications the impact actually of the molecular tumor board suggestions, which really have an impact on the overall survival of patients. Now, our vision and ambition is that we would like to have those patient organoids a seat at the table. So on top of the molecular tumor board, bring in patient organoid data to further improve the prediction and further be able to tailor specific drug treatments during clinical trials and later on. Now, very quickly, we're also looking into genetic predispositions and how we can use that and recapitulate that using in vitro systems. So you certainly have heard about an HLA association with, for example, drug-induced AEs or also anti-drug antibodies and other things. And this is something we have been
Adrian, you got one more minute, please.
Yes, one more minute.
One last minute,
we're
please.
Working,
yeah. Thank you.
Yes, we're working currently with a team in Cincinnati building genetically predefined liver organoid system to be able to address genetic predispositions for drug-induced adverse events. So in order to be able to closely monitor those patients, in order to think about potential co-medications, alter the dosing, or in the worst case also exclude patients. And with that, I'm at the end. So this is what I tried to convey today, kind of bring the safety model next to the efficacy model into the world of patient-derived tissues to assess an individual therapeutic window and address the individual benefit-risk ratio. We alluded to that in a couple of recent publications. And I am sure you have seen also that Roche has made a clear commitment to adoption of these types of models by the recent opening of the Roche Institute for Translational Bioengineering. We also alluded to that in a few recent publications in Science and Stem Cell Reports. And with that, I've come to the end. And I hope that was interesting and relevant, and I'm happy to take any questions. Thank you very much.
Yeah. Excellent presentation, Adrian. We are running short of time, but we have time for maybe 2 or 3 crisp, short, to-the-point questions.
Thank you very much, Adrian. Excellent presentation. My first question is, I'm ex-BMS and I have used anti-CTLA-4 ERVI that you referred to. The initial days of clinical trials produce very adverse reaction where the tumor size would go really big and then cut down. Don't you think these models would miss out on such kind of reaction, number one? Number two is, from the regulatory perspective, what's your guess on the acceptability of these testing methods, MPS testing methods, to personalize the medicine? Like, do you need to do clinical trials to get approvals regulatorily, or you can just go and collect any patient sample and tell you are not going to be efficaciously treated by this particular drug? So 2 questions. One on ERVI and the second one is on the regulatory. Thank you.
Yeah, thanks a lot. Excellent questions, very relevant questions. So of course, these are challenging. So to your first question, these are very challenging adverse events to be captured. I wouldn't say we can capture all of these adverse events, but at least some of them, specifically those on-target, off-tumor effects, we actually do have quite nice data where we believe we can capture them. Not in all cases, but at least to improve the situation, and certainly better than compared to what we have been using and assessing before. We can capture now effects that were not seen in, for example, cyno studies before. To your second questions, we are now currently at the stage where we introduce those patient-derived tissue models as part of a personalized medicine program in clinical trials. I think before we go into really clinical practice, we would like to start with our clinical trials and see how's the translational aspects, how well does it work. We need to think of all the operational aspects and also get a bit more confidence into the data. But we are aware and we are in discussions with teams around the world in large clinical academic centers actually that do, as we speak today, run quite large trials where the ambition is to take actually decisions or at least support this clinical decision making for those patients based on data including patient-derived organoid data. So I hope that addressed your questions.
Yeah, we'll take Manny's question and then one last question from the students, please.
Yeah, very good presentation, Adrian. And my question relates to the immunotoxicity of drugs. We have probably industrial pollutants in the 1980s have shown the immunotoxicity, halothane, bromobenzene, quite a few things that people have investigated a lot. And then drugs, I mean, you're talking about carbamazepine, dial-epoxide. Most of them covalently bind to the proteins, and that triggers the immune activation system because of the hapten protein activation. Pharma companies for the last 30 years, whether Merck, J&J, or BMS, or anybody else, first thing they do in drug discovery screening when a compound is going to be nominated as a candidate for preclinical development, they do screen for these reactive metabolites to ensure they won't get this kind of immune activation systems. So I'm just wondering, how do you couple these things to your model today that you're trying to look at? Apart from the HLA activation, I know those are all also part of it, but this is the triggering initiative process. Can you please explain it to me?
Thank you very much. Yeah, excellent question. What I can say is we're working right now on exactly these questions. Yeah, you seem to be well aware of what's been done in a preclinical assessment. You know, you can measure reactive metabolites and all of these kind of unfavorable properties that you don't want typically for, in this case, small molecules in drug discovery. Sometimes you do move forward with molecules that have such flags, which bears the risk of generating danger signals and generating hapten so that the immune system all of a sudden then in a particular clinical situation starts to become activated and we have an adverse event. It hasn't been possible, at least to my knowledge, so that teams have been able to move all of those things together in an MPS system and really measure it. We're currently working on it. It looks quite promising. It's relatively complex, but our first data look quite promising that using MPS, we're actually able to do that. I hope next time that I'm presenting either at this event or another one, I'm able to share such kind of data with you. But it's right to the point, your question. Thank you.
Great. And one question from students, anybody you have? You guys are hungry? Okay. All right. Adrian, beautiful presentation. Thank you so much. And, look forward to other presentations from you. So we'll now break for lunch, Anushka, and be back at 1:30 sharp. Yeah. Thank you very much.
Good. Thanks a lot. Bye-bye.